introduction, and thank you to the organizers for inviting me to be part of this panel. I'm very much fitting along the lines of Rachel's talk because I'm going to talk about closing the scientific gap by recruiting individuals underrepresented in current research, and by that I mean early onset Alzheimer's disease. Early onset sporadic Alzheimer's disease is defined as disease onset before the age of 65. Oftentimes these patients are in their 40s and 50s, they're gainfully employed, they're raising their children, they're definitely not yet ready to retire. Contrary to the perception that this is a rare disease or a rare variant, there are 200 to 300,000 cases in the United States. If we were to compare that to the second most common early onset dementia, frontotemporal dementia, one can easily appreciate how much more common early onset Alzheimer's is. There are only 20 to 30,000 frontotemporal dementia cases in the US now. There are important distinctions relative to late onset Alzheimer's disease, which makes this an important cohort to study. First and foremost, there are many non-amnestic presentations. The majority of the cases actually present with posterior cortical atrophy, language variant, frontal variant, many non-amnestic presentations as compared to late onset, where amnestic presentation is the norm in 75% of the cases. In addition, early onset Alzheimer's disease is a very aggressive disease. The rate of progression is much faster compared to late onset, and you can see the bar graphs on the top in quadrant 1, where in blue you see the annualized rate of progression in late onset, and in green you see the annualized rate of progression on MMSE in early onset. There is 5-fold greater, more progressive change on MMSE score in early onset compared to late in the UCSF data. And in addition to that, they're young, so they suffer from fewer medical comorbidities and from fewer diseases of the aging brain, such as TDP-43 vascular pathology and so forth. Now to quadrant 2. A very common misperception about early onset is that that is predetermined by the 3 autosomal dominant genes, IPP, presenilin-1, and presenilin-2. And you can easily see that the pie chart shows only 6% of early onset cases are actually due to autosomal dominant mutations in one of those 3 genes. Thus, early onset Alzheimer's is likely enriched for many novel AD-related genes. Many cases are familial, and the genetic risk factors beg to be discovered. And also, because it's so heterogeneous and has these atypical presentations and extreme variants, it's a very good model to study genotype-phenotype relationships. We just now embarked a new project called the Longitudinal Early Onset Alzheimer's Disease Study, or LEADS, which I'm co-leading with Gil Rabinovich, Brad Dickerson, Maria Carrillo. It is a U.S. study with 15 sites. We are set up to enroll 400 amyloid-positive APP, presenilin-1 and presenilin-2 gene-negative early onset Alzheimer's disease patients and 100 controls, and to collect detailed cognitive, clinical, and biomarker data, including all types of imaging modalities and fluid biomarkers and whole genome sequencing, RNA, and IPCS. This is a public-private partnership, and we're very thankful to our partners, the Alzheimer's Association and the industry, and are always looking for more modeling by ADNI's model. In addition, we're collecting the biomarkers with ADNI 3 protocol in order to be able to compare to the late onset cases in ADNI. What are our goals? Obviously, this is an understudied population that is often misdiagnosed, or diagnosis is terribly delayed. We would like to improve on that, and we would like to know which exact biomarkers and imaging markers are characteristic of the disease progression across the spectrum that is seen, of presentations that is seen in this disorder, and also develop sensitive clinical trial outcome measures for this population. We also, of course, are very interested in finding new genes within this sample, but also enlarging it, expanding it to other early onset AD DNA samples. Very importantly, this is where precision medicine comes in. We will do a deep multi-omic molecular profiling of these cases to characterize the rare subtypes, to follow them along, to develop biomarkers and understand mechanism of disease and point to new therapeutic targets potentially. And last but not least, we're establishing a network of sites that could become clinical trial sites for this unique population with pure AD pathology, but very rapid progression. Thank you.